Let's go ahead and get started. I'm Dean Jones from Emory University, and I'm going to act as the moderator for this session. We're going to have a panel discussion addressing issues that we covered this morning and really opening up to give some other perspectives. We have a distinguished panel. You've already met some of them, but the one we're going to start with, some comments from Susan Sumner and Andrew Patterson, because they have not spoken already, and then we'll go on and open up for just a more general panel discussion. So I'd like to start with having a few opening comments from Susan Sumner. Susan? Okay. And you have to turn on your microphone.
There you go. Thanks, Dean. And I think we're going to go, I'll try to stay within this 5 to 10 minute range. Okay. So is that okay? Okay, so I did want to start by saying I totally agree with all the previous speakers about their pros and cons of the targeted and broad spectrum methods and the techniques, even when they didn't agree with each other, because this is pretty complicated and there's lots of ways to approach the problem. And opinions can differ and they can be correct. So I appreciate Erin's work and using the ion mobility to get specificity of compounds, and we've been doing that kind of work with endocannabinoids also and using it in a more targeted approach to really try to pick up things that are very difficult to pick up in these broad spectrums. So there were a couple of people that mentioned NMR for screening, and we're really fond of this, particularly in studies where epidemiologists talk about their precious samples and they want them back. So we can use the NMR, it's non-destructive, we can give those samples back, and we can get a lot of information out of NMR metabolomics, and then go on and do some targeted methods or methods for environmental measures as well. And I wanted to add to the conversation on the data matter that most of our studies are focused on comparisons among cases and controls. And in our comparisons of cases and controls for health outcomes, most of the compounds that differentiate our cases and controls are endogenous compounds that map to biochemical pathways. We do get drugs. We'll find out that the cases had some drugs that the controls didn't. Or maybe we'll get something that comes out of eating sugarless candies, like maybe from a diabetic study, so we know probably where that's coming from. But I've never done the study where I've seen the pesticide differentiate my cases and controls, or the herbicide. So in our normal metabolomics workflow, that's not what we're seeing. And so it really speaks to this importance of having these targeted methods that are going to measure those low levels of herbicides and pesticides to be able to utilize that with our metabolomics investigations. So Dean asked me to diverge a bit from the conversations about the detection and go on to talk about metabolomics and memory. And I think this is because we did a study in 2006. It wasn't published until 2009 because I think people didn't believe it, and metabolomics was relatively new then. But we took pregnant rats and dosed them with butylbenzyl phthalate, and we collected urine and tissues from the offspring at 26 days after the delivery of the offspring. So butylbenzyl phthalate is long gone, okay? And at 26 days after that. And we saw differences in the urinary metabolome of the offspring that were born to the dams that had the butylbenzyl phthalate exposure compared to the offspring born to the dams that had the vehicle exposure. And we saw those differences in urine. And we also looked at target and non-target tissues of the compound, and we saw the differences also in the target tissue, but not in non-target tissues. And so I just wanted to speak to the fact that exposure is something that can pass, right? So we may be wanting to focus on the memory of that exposure, and what does that do to endogenous pathways, and how can we use that information to reconstruct prior exposure. So we had submitted several grant proposals on using metabolomics to assess prior exposure and prior utero exposure, and so far those have all been rejected, but I'm hoping maybe maybe these people will be reviewers on my next submission. So I also just wanted to say that we've been working on a number of studies with cord blood that may be of interest to this group. And I'm thinking of cord blood as now being the exposure, right, to the fetus. Because if you have a mother who has a high concentration of arsenic in her urine and another group of mothers with low concentrations of arsenic in their urine, we see differences in the metabotype of the cord blood. Now, those differences in the metabotype of the cord blood aren't environmentally relevant chemicals that we're noticing. They're endogenous compounds. So now we see that in utero, and these are in human subjects, that the in utero exposure is different. So that's the in utero exposome, and I'm really excited about that. So we've done some work with Rebecca Fry, who's at UNC Chapel Hill, analyzing samples that she has with arsenic. We've also been working with Frederica Pereira, and who's at Columbia, and I'm sure you know of her work in molecular epidemiology, and we've been looking at exposure that's been well-characterized, well-defined exposure to PAHs, okay, because that's what they're world-renowned in doing. They're also world-renowned in measuring the cognitive development in the children, and this was done at maybe, don't know without my notes, 12 months, 24 months, and 36 months. And so they're looking at exposure and this health outcome. So we've been looking at the metabotype of the cord blood from those same children. And now it's really complicated to try to analyze the data because we have all these different exposure parameters. And which ones are associated with this cognitive delay, and then, or development, I'm not sure I'm using the right words on that, and then which one's associated with the metabotype. So using the normal metabolomics workflow, we haven't really been able to unravel that, but we've started using something called structure equation modeling. And structure equation modeling enables you to put in lots of different informations about exposure and then be able to model that against different outcomes simultaneously. And I'm not the mathematician, but that's how they told me to say it for you. But that seems to be working. And I just wanted to say that, you know, I think that we have lots of really good data and really good information on exposure, but we need better ways to model that data. So, and then David had mentioned that I should say a few words about resiliency and what we are looking at. And you know, we've done some studies, and a study I was telling David about last night is with Pat Brophy and David Ashkenazi, who are medical doctors who study acute kidney injury in neonates. And so we did this case control study in the neonates, and one of the biomarkers in the cases was hydroxybutyrate. And I was like, yeah, I remember hydroxybutyrate being elevated in studies in kidney disease. So I went to look up a couple of papers on that. And I laughed because there was this paper published in 2014 on the protective effects of hydroxybutyrate on paraquat toxicity in the rat kidney. So now I'm thinking, wow, all these studies where I've seen hydroxybutyrate elevated in kidney disease, it may be a good thing. It's resilience. It's a protective mechanism that kicks in. So we have to be really careful about how we're interpreting the data. When we see something change, is it a good change or is it a bad change? And what's resiliency and what's adverse response? And how are we going to design our studies to get to that? So I'm very excited about those issues. And even when it comes to chemical exposure, you know, if you measure arsenic in the urine, is that a good thing or a bad thing? Well, to me, that had always been a bad thing. That's what I was taught, right? But there was a paper published that actually came out of Berkeley in 2014 where they found a decreased breast cancer incidence and mortality in communities with high arsenic exposure compared with communities with low arsenic exposures. And it's a very interesting paper, and they go on to do in vitro work with arsenic with breast cancer cell lines to demonstrate a significant reduction in cell viability and increased apoptosis with the arsenic, okay? And so the investigators from Berkeley go on to explain that maybe we shouldn't have been too surprised about this because arsenic's being used as an effective treatment for certain types of leukemia. But I think that this paper is really important because, you know, not that I'm proposing to go and expose people to arsenic, but we really have to understand the relevance of the data that we're generating and what it means. Okay, and I'm going to take one more minute, and I just wanted to say that we've also been looking at effects of early life exposures to antibiotics on, and these are in animal models, and we've been showing, and this work's been conducted with Martin Blazer, who's at New York University Medical Center, and this is where we're looking at the metabolomics profiles of mice that either have had normal vehicle or had low doses, subtherapeutic doses of antibiotics, and the ones with the subtherapeutic doses of antibiotics develop an obesity phenotype, and they have differences in their metabolome. Interestingly, if you take the feces from those vehicle or the low-dose exposed animals, you put those into germ-free mice, you also get differences in the metabotype. So this goes to what's going to be this sort of multi-generational effect of these things, which I think a lot of this stuff from exposure, and Carol and I were talking about this earlier, is really going to be derived from a really good understanding of what's going on in the animal models and how are we going to translate that to humans. And then the last thing I wanted to mention, because I always get a little bit interested in these symposiums when we're talking about metabolomics, and I do want to say metabolomics is my big love, so I never want to say anything bad about that. But I don't want to forget that we do have an exposure dose response and susceptibility paradigm. And I know when I go to Duke and NC State and UNC and I give presentations and classes there, it seems like the students aren't being taught that exposure dose response and susceptibility paradigm anymore. And I think that that's a shame because we really put a lot of effort in that. And I know many people in this room who are kind of laughing, we're part of that. And what I think is that metabolomics is a great opportunity to look across the entire exposure dose response paradigm and not limit that to this case and control kind of area, and really try to understand how do perturbations in the levels of DNA methylations or DNA adducts or DNA repair capacity compare with perturbations in your metabolome. So I think all of these things wrap up to the exposure, and we need to be able to model it. Okay, thanks.
Thanks, Susan. Okay, let's have a few words now from Andrew Patterson.
Sure. So I just wanted to add to some of the discussions earlier. A lot of these concepts remind me of an idea that was introduced in the 40s and 50s regarding biochemical individuality. So this is something introduced by, I believe it's Roger Williams at the time, and this is the idea that it's very difficult to kind of put persons into a very defined set of parameters. And I think through discussions today, but also following the literature, the metabolomics literature over the last 10, 15 years, it's really started to kind of exemplify that this is what this is all about. So what are these things that are contributing to this? Well, obviously genetics being one, but microbial populations or differences in those populations have now started to be a big contributor to this individuality. And also, of course, kind of as Susan pointed out, this idea of the metabotype. And one thing that I think is important to address that was kind of touched a little bit by Anthony's talk is study design. And so how do we really get at this idea of individuality very carefully? Well, I would argue that it's very challenging to do so in just a kind of a snapshot fashion where we're taking a sample on a certain day and considering that to be representative of what you look like over a period of time. I mean, I know I go to the doctor now and you can plot out my results for the last 10 years and I can see trends in certain directions one way or the other. And I think this is an incredibly valuable way of looking at your health and disease progression. So with study design in particular, I think this idea of Toby talked a little bit about is longitudinal studies where you have repeated samplings of people over a long period of time, which would really help you to establish what the baseline is, not just for, say, exposure, but also more so for what you look like on a day-to-day basis. Maybe you just went, contributed a sample at the wrong time. And I think this is one of the complications that we face when we try to do cases and controls, because we're comparing people at a single time point as opposed to, say, a time that's much more representative of what they look like over, say, a week or 6 months. Who really knows what the answer is? So we're starting to investigate some of these studies, looking at repeated sampling of people over long periods of time to see, in fact, how they do vary. And of course, you can see days where they took Tylenol or they drank some tea, whereas opposed to other days they didn't. And so these are the things that are all contributing to this idea of this biochemical individuality. So that's what I really want to say.
Okay. Thanks, Andrew. So I think what I'd like to do now is progress with a few issues that are broader in nature and that really impact on a lot of the things that we've heard about this morning. And these will echo some of the things, but I think hopefully amplify some of the issues. And one was that discussion of the reference metabolome, and this is really perfect following from the comments on the human individuality, that ultimately we have to be able to deal with metabolomics at an N of 1. And so we have to have some way to bring that back into the population. So comments from the panel on how we can move forward with that concept of developing, really building upon what David Wishart has done with the Human Metabolomics database, but now bringing that into a very workable reference metabolome. Comments on that?
Well, I think David's done an excellent job in that area of coming out with, what's the metabolome of saliva or urine or blood, and doing all the curation, right? And the analysis associated with that. So I think that area is moving and growing and really important, but how do you know what the variability is from that, right? So there was a paper published by Shure last year. I hope I say that right. I think it's Shure, that's doing a correlation of SNPs to disease outcomes and then has metabolomics profiles. And they created the metabolomics profiles out of the twin study in the UK and another large biorepository that I can't remember the name of because I don't have a slide. But anyway, so what they were looking at, what's the variability between twins in the SNPs and in the metabolites, right? And then what's the variability between one twin and the rest of the population to look at what's the genetic inheritance. And then they look at SNPs associated with environment, what's the environmental inheritance, and then what's between that. So I think eventually we'll get to individuality, but I think we have to be able to correlate it back to what we know. And I think all the work that's been done in SNPs is really important.
But from the exposure standpoint, we have a special challenge. And as we have NHANES data, for instance, so we have a lot of available data, but the human experience is changing. So if you take archival samples that were collected 30 years ago, they're different from what they are if they're collected 10 years ago. So one of the challenges, and so how are we going to deal with that type of an issue with regard to environmental exposures, even food exposures for that matter, because the food processing, the products that we get are changing. Come on, panel. That's our job.
Yeah, I don't really see the point of having a standard person for the metabolome. Maybe there's something I'm missing, but people are different.
People are different, so
it'd be nice to know what the variability is of the metabolome across population at different times, and that would be interesting. But coming up with a standard set of blood or a standard set of urine and working out the metabolome, and unless you want to do round-robin tests at different laboratories and try to standardize methods, I don't really see much point in it. But maybe I'm missing something.
Well, maybe I'm missing something, but aren't we going to need to be able to standardize things between collections that are in Philadelphia versus ones that are done in Berkeley, and have some ability to use that information to try to understand differences, geographic differences in terms of outcomes? It seems as though we have to have some way to reference things.
Like I said, maybe I'm missing something. I mean, what you're talking about is the variation of metabolome across populations separated geographically. It could be separated ethnically. It could be different dietary factors. There's all kinds of things you could think about, and that would be interesting to look at the variation. But to try to come up with one standard metabolome, I think, is not really very sensible.
Okay, well, comments? Yes?
Yeah, I think I agree.
Microphone, yes.
I agree with Stephen. I mean, in an epidemiology study, the means don't matter, right? You're looking at variation from person to person around the mean. Go back and look at what your regression program does, and the first thing it does is it tracks the mean from every variable, and then it does the calculations, right? It's the variation from measurement to measurement in predictor and the variation from measurement to measurement in outcome, right? So if people in Philadelphia are different than people in California because they eat a different diet, well, you need to collect dietary information so that you can capture that, and Haynes does, right? And cohort studies generally do. If they're different for other reasons, you measure those. I mean, that's what the exposome is about. We want to measure all these different exposures and see how they explain variations in the metabolome. If your point is that I collect data in Philadelphia and analyze it in a lab in Philadelphia and collect data in Berkeley and analyze it in a lab in Berkeley, then I would say your study should never be funded, right? I mean, you collect the data all over the country and you analyze it in one lab. That's the fundamental of doing any kind of epidemiology study. So I don't think that we're going to get
well, I guess
the problem that you're trying to solve.
Well, but the problem that we're trying to solve is we're trying to understand your exposures and your health, N of 1. So if you have N of 1, you don't have a cohort study.
I have an N of 1, I don't have an explanation either.
So you're saying the whole process is futile when it comes to any individual information.
You cannot attribute causality to anything in an N of 1.
Okay, given.
So I'm not interested in an N of 1. I'm interested in doing a study and finding out what metabolomic profiles are associated with what exposure, or what metabolomic profiles are associated and seem to be predictive of what health outcome.
Okay,
right?
so
but I'm not drawing a conclusion about you.
But ultimately, if you're not going to be able to get information about an individual, is there any purpose at all?
Of course. Nobody in their right mind would tell you that you're having hypertension or not is going to change your risk of having a heart attack. Right? There are plenty of people with high blood pressure who live a long life and never have a heart attack. The only thing that people will ever tell you, if they know what they're talking about, is that on average, people with high blood pressure have heart attacks at a higher rate. But you might be one of the ones who's resistant.
So what's your reference when you do that?
The reference is the Framingham Heart Study, which found that on average, people with higher blood pressure had more heart attacks.
So you have a reference. You have a reference.
No, because they looked at variations. My reference is, there is no reference. The variation in exposure is associated with the variation in outcome. But that's not predictive of an individual. It's predictive of a population.
Okay, well, so we probably have talked about this enough, so let's move on. But I guess, it's just one of those, this illustrates the differences in thinking about things, because when we go to precision medicine or personalized medicine, we usually have a reference. And for the exposome, the question is there going to be a reference? And, one of the answers is no. So, okay, go ahead.
Albert Donne again. From breath research, I want to share with you that over the past 20 years, researchers have learned that in order to get individual references, because the metabolome changes on a diurnal cycle, if not other cycles, it's absolutely critical to standardize the time of day of sampling and measurement, as well as, of course, time since eating and drinking. Just consider body temperature. We know there's one good time of day to measure it before you get out of bed in the morning, and everything after that is worthless. So in a similar vein, because we have natural cycles of increased or decreased metabolism during the day, it's critical that we collect and study samples among individuals and even within the same individual at the same time of day.
Okay. Well, we've heard about longitudinal studies. Maybe Andrew could talk about that just a little bit as far as that inter-individual variation, because that's a different way to reference. If we had information on an individual, then they become their own reference for the future.
Yeah, sure. I would like to make one point, though. I think the idea of building a reference standard for the population, I don't agree is maybe as valuable. However, I would argue that indeed, trying to annotate that as many chemicals as possible in that is incredibly valuable. So I think the more knowledge we have about a particular sample, whether it's representative or not, I don't think is what we're trying to fight for. What we need to do is build more or annotate more than where we currently are. And so, efforts by like David and others, is incredibly valuable, because this does tell us what is in a potential sample. It doesn't necessarily have to be representative of a population. So I think that's really important. So yeah, the question. About longitudinal studies. Yeah. So I think very importantly that it's better often having these references based on the population. However, it's still important to always be looking at the individual as their own control throughout a particular study or throughout a particular time frame. I mean, I think you can learn incredibly, a valuable amount of information through this type of approach, because it does take out some of these weird effects associated with, say, dietary changes that may be associated with a particular population. That maybe you don't eat fish, but others do, and this may be a huge hallmark of what your metabotype is, just due to, say, the presence of TMAO or something like that. So, I'm incredibly in favor of that longitudinal approach where individuals are their own controls.
Yeah, and Susan, I think you had a comment on that.
Yeah, in a large epi study in the right population, I think this longitudinal approach and collecting those samples at the same time of day is really a wonderful thing to have. There are other kinds of studies that we do with metabolomics where we are working with, sick people, babies, that urinate when they're going to, and they're going to eat when they're going to. So we had to kind of work around that. And one of the things is if the health outcome phenotype is significant enough, the variability that you have in the time of day a sample's collected or when you've eaten or when you've not eaten is going to not emerge as your biomarker, because it's going to be variable in your cases and in your controls. And so while I agree that there's the ideal world of sample collection, we're not always working within that ideal world. And as a matter of fact, we seldomly work within that ideal world. So,
Okay, thank you. I'll change direction to something that Steve had mentioned. No, Steve wanted to. Sure, go ahead. Yeah.
This idea of longitudinal studies is related to the fact that the metabolome is comprised of thousands of small molecules, most of which turn over relatively rapidly. That's a disadvantage for an exposure marker, because it leads to within subject variation, which leads to measurement error, exposure measurement error in trying to assess a exposure response relationship or to find differences between cases and controls. So that's an inherent problem with using metabolomics for exposure studies. But having said that, there is variation in the residence time of biomarkers. Some of them stick around longer than others. Some of them are generated daily from endogenous processes or because the dietary factors remain fairly constant over long periods of time, so they can still pick something out. If you do a study and compare cases and controls, this measurement error associated with changes in biomarker levels over time attenuates the relationship. So you make it less likely you're going to find a difference between your cases and controls. So you're going to have a lot of false negatives, but you're not going to have a lot of false positives. So if you do find significant differences after false discovery correction, those things that you find, those small molecules, are really worth following up because they're very likely to be associated with something that's interesting. So I think we have to keep that in mind. And I agree with Andrew that if you could get longitudinal data, it would allow you to reduce this measurement error in a systematic way and even adjust for it in some studies. So that would be useful. But getting longitudinal data in epidemiology studies is very expensive, very difficult, and you're not going to find a lot of them that will be available.
Okay. Yep.
Yeah, so I think the original intent of your question was personalized medicine. Can the exposome get us there?
Personalized exposures is what I was really
Personalized
thinking about.
exposure.
If you're 1 in 100 who's exposed to something, how are we going to get there?
Right. So I guess, and it's just kind of taken a while for me to kind of formulate this, but we have to start from disease first at a population level and measure the exposome in these case controls and begin to understand what are significantly associated, hopefully new biomarkers can be identified for various diseases that are significantly associated with these various diseases. So basically disease first, collecting lots of information in case control type studies to evaluate and annotate as best we can the exposomes in these various models. That can begin to give us a population reference, possibly. And then from that, we could use the probabilistic information saying, okay, we know that in disease X, this particular set of chemicals are elevated or associated with that. Therefore, you as an individual who are showing these same elevations or decreases or whatever it might be, have a higher probability of possibly succumbing to that particular disease. So high blood pressure, you have a higher probability of heart attack, but you may never get one. So again, so I think it begins with disease first, case controls, understanding these new and novel biomarkers, and then we can take that population information and say, okay, at the individual level, there's a probability that you may succumb.
Yeah. So I'm going to challenge the panel to another question, but I'm expecting people to get up to that microscope or to the microphone, and I apologize that I didn't invite you up to the microphone before. So please, if you have questions that you would like to be discussed. But following up on that question is, how many people would we need to do? Because if you're locked into a case control, the current studies are relatively small, and how large of a study do we need? And then coming to one of Steve's points earlier, how do we scale up, in terms of getting large enough studies? So are there thoughts on that? How large is large enough?
So I'll weigh in on that one. So being from a proteomics core lab, we did a study with over 6,000 patients that all had sarcopenia versus control. And it was great doing the 6,000 patients, but the bioinformatics was a nightmare to analyze and try and understand the differences and the different biodiversity and stuff. So I think what we learned from that is that we started with a 200-person study, and we were able to make conclusions fast. And then 2 years later, we've finally been able to make conclusions about the 6,000-person study, but it depends on how fast you want to make conclusions and how fast you think the informatics is going to be able to do it, because if you get too big, the normalization and everything is just so difficult.
Yeah, I'll just add a quick word. It depends on the size of your effect. So if you have a very large effect, like let's say you have a 100-fold difference in some metabolite level between cases and controls. You're not going to need a very big study to find that. If you've got a 1.02 fold difference between the two, you're going to need an extremely large study to detect that. So it goes back to basic kind of power calculations and the difference in the means of the two populations that you're comparing.
So, okay. Any other comments on that? Let's go ahead with comments from...
I'm rishabh. I'm with the Navy Marine Corps Public Health Center, and therefore have to say that all the comments I'm making today are my own, and they do not represent the opinion of the Department of Navy or the Department of Defense. And actually, the question I have fits well into the recent discussion because it's actually two questions that are correlated. And the first one is that now that we've defined the exposome as all possible stressors during the lifetime, and that we've been talking about the utility of a non-targeted metabolite analysis, my first question is, given those two parameters, how do you define a priori your control population? And I would say then, as the correlated question, if you're going to define, as is often done in case control, your control population as those people who do not have the disease, then what we are missing is perhaps the most informative part of the population, which is the people who had the exposures that really are important, even if we don't know them, but did not respond to the disease, because that's exactly where, when you have a small effect, or when almost everyone responds, you'll get the most information about the adverse outcome pathway or mode of action, whatever you want to call it, from the people who have most of the characteristics but are missing one that is necessary for the disease.
Comments on that?
Yeah, so I like those concepts very much, and definitely we can control for that when we do animal studies, and we can see animals that develop the adverse response or don't develop the adverse response under the same exposure conditions, right? So we can get information for that.
Yeah, that first assumes that you know what might cause it, and it doesn't address the problem we're really dealing here with is multiple exposures, and most of them having to do with things like lifestyle instead of chemicals. I've been dealing with chemical mixtures for a long time, decades, and the potential toxicities. And we actually have loads of ways of modeling and dealing with those. But once you throw in other things that we don't have easy measures for, including another field I've worked on, which is radiation, you're suddenly making it very difficult. We can do the experiment for radiation on animals, but we can't do the radiation plus the chemicals plus the viruses, which, by the way, are different for animals than they are for humans. For the
Absolutely.
same effect. So,
and I'm not disagreeing with you. I think that you're absolutely right about that. And that's where biorepositories with well-documented metadata is what we really need, because this is what I was referring to earlier about the need and improvement of modeling the data, that if we take our normal workflow and metabolomic study and try to link the phenotype of, bad air quality in the city versus another city with good air quality, and you see differences in the metabotype, that might not mean that much, because there also may be differences in nutrition in those two cities, or whether or not they have exercise, have sidewalks and get more exercise, or live in rural areas, or So I understand where the concept is from, but I think this comes into the whole thing of how we need to make sure that we have that metadata and that we have tools for being able to model all of that data together
without violating personal privacy.
Yeah, thank you. And I want to come to your question in just a second, but let's follow up on that with this issue of chemical space and the conceptual grid concept that Christopher Wilde originally proposed. And I want to come back to Anthony's comments on that, but let's take this question or comment from the floor first.
Okay, so I agree that cohort studies are extensive, but I've heard enough about case control studies here to make me nervous. Okay, so I want to tell you why, as an epidemiologist, I get nervous when I hear the phrase case control study, because a case control study gives you a valid inference only if the controls are representative of the population that gave rise to the cases. And that's because, in fact, the purpose of the controls, we don't care about them as people, right? The purpose of the controls is to give you the distribution of exposure in the population that gave rise to the cases. So you can compare that to the distribution of exposure in the cases and say, they look a little different, don't they? Right? That's what you do in a case control study. Now, if you start with a cohort study and do a matched case control study, then that assumption, I mean, assuming you're not an idiot, right, that assumption is going to be true. The controls will be representative of the population that gave rise to the cases. In most other scenarios, it's not true. You know, you can't do random digit dialing anymore. No one answers their phone, and a third of the population only has cell phones, which you can't call, right? You're not getting a representative sample, right? You know, hospital controls, give me a break, right? The people who came in with broken legs are not representative of the population that gave rise to the heart attacks, you know? You go out and you try to recruit people, you know, I mean, I've gotten upwards of 25% of the people that I've tried to recruit as controls in a case control study of heart attacks to actually agree to participate in the study. Are those 25% representative of the other 75%? I mean, if you believe that, have I got a subprime mortgage for you? You know, so it's gotten very hard, and it was never easy to, in fact, get appropriate controls. And so it's really important, even if we're doing case control studies, to do nested case control studies within cohorts, because otherwise, you know, if the metabolome in apples is different than the metabolome in oranges, you know, so what? Just tells you that apples aren't oranges. If you want to discover whether some exposure produces a change in metabolome in oranges, you better compare them to oranges. And most case control studies don't.
Thank you. So can we come back to the this question of chemical space that Anthony brought up, and how are we going to approach the problem of the conceptual grid of the exposome? This is in the original Wild paper, he talked about this developing a conceptual grid, and we have chemical space, and that's probably the closest we can come. So how do we think about that in terms of developing a exposome research structure, if you will, for doing this? Any thoughts on that? We have one of the world's experts right there in the middle. Anthony's expert on the analytics in terms of chemical space.
Yeah, I guess, you know, from the soup of chemicals that are out there, we need to differentiate what are the important ones. We're all exposed to tens of thousands of chemicals, you know, throughout a month, throughout a day, throughout a lifetime, what we're exposed to. And I think using the exposome paradigm and measuring from, you know, starting with prospective cohorts and just kind of collecting blood over time, samples that we can compare, nest, and use these nested case controls from therein, we'll start to tease out what of this soup of chemicals are the important ones. So there might be 200,000 that we want to measure, but it may turn out that there's really only 2,000 that we really should focus on. And that might be teased out in time by using kind of this disease-first approach and the exposome and trying to measure as much as we possibly can, and understanding, annotating what those things are. So then this space becomes much smaller and much more manageable. But, you know, to just say we're going to measure everything all the time is, you know, it's impossible and it'll never be done. So I think it's one of the reasons why I was drawn to the exposome concept, was that if I were to use this and, you know, have cases and controls where I could measure differences and find significant patterns, that those chemicals might be the ones I would focus on. So instead of measuring as much as I possibly can in the air and water, and that's great, we've done it for 40 years and it's gotten us some information, it may be better to measure the 2,000 things or so that are in us often. So I think that might be the way to kind of look at it,
Okay,
if that makes any sense.
Yes, Erin?
Yeah, I don't know if you guys have looked at Kim Anderson's work, where she has the rubber bracelets everybody wears around and it collects the PAHs. So she, like all the kids have the college bracelet, and then she collects them and washes them and then sees what PAHs they were exposed to that week. I think that's a really interesting idea of like maybe prioritizing what people are exposed to, because maybe it's not, like the half-life is too fast to measure it in the blood, but maybe we can figure out some way of detecting it exterior and then focus on it interior. I don't know.
Okay.
Hi, Hanaka Troy from University at Albany. I was wondering about the reliability of the internal dose as it relates to the within-person variability versus the between-person variability. So we've seen that, to use PAH as an example, we've seen that the within-person variability in New York City for PAH exposure is so much higher than the, let me see if I'm getting this right, than the within-person variability in Krakow, Poland, where the median concentration of PAH is about 100 times higher. So, I mean, within a given time point, the between-person variability in Krakow is much lower than the between-person variability in New York City. So that makes me worry a little bit about the using, like it's just a single blood spot or blood sample as a marker of internal dose. And I wondered whether you had any thoughts about external environmental samples as standards or reference.
I couldn't really see you very well. Were you looking at me? So I'll
Yes, definitely,
try to answer
Steve.
the question. This question of within versus between subject variability is the heart of being able to establish a dose-response relationship or to find a difference between diseased people and well people. It's not particular to metabolomics. It applies to external measurements of pollutants. It applies to anything, questionnaire data, and so on. It is possible in a single sample to find significant differences that do discriminate for disease if there's a very large effect. For example, you have a biomarker that is only present in smokers, like cotinine. If you measure cotinine at a reasonably high level in a person one time, it can tell you almost certainly that person is a smoker. Cotinine is a small molecule, so it's just an example of how metabolomics, where you measure thousands of things, would also allow you to find certain biomarkers that are going to be able to discriminate for either population status or disease status or exposure status or something else. The reason we're going to metabolomics or to internal measures is because they integrate things from all sources. You can measure external exposure. We've been doing that for a long time, as Anthony said. And I think we've gotten about as far as we're going to go. So the next big discoveries of disease, I suspect, and this is my opinion, are going to come about because we're measuring lots of things inside people, and we're not going to rule out endogenous, food-derived, drug-derived, inflammation-derived markers that are perhaps driving a lot of these chronic disease processes.
Are there other comments from the panel on that?
I would just like to add one important observation that's kind of come to light in the last year or last years, and that's the work by Peter Turnbull showing that it's not just host genetics that seem to be driving a lot of these changes as well. It's bacterial populations that seem to be a major contributor, and his work with digoxin is a prime example of that. And I think this is an important consideration for these types of studies. However, as you just said, the metabolomic approach really does capture this information. It doesn't matter if it's host metabolism or bacterial metabolism. You see this reflection in that profile. So, from that standpoint, it's a very powerful approach.
Okay, good. Next.
Thank you. Joyce Suji, I'm with Exponent, and I'm part of the committee. I think some of the biggest challenges ahead are going to be analytical and in sample handling. As you look at more and more compounds, and as you try to get better detection limits. I mean, right now with dioxins, to get decent detection limits, you need about 80 mils of blood. That's really hard to do on kids. If you look at NHANES data, most of the arsenic species are non-detect except for maybe DMA. So clearly we need to try to get better, cheaper, more rapid ability to measure chemicals. And also, when you're sampling for multiple chemicals in blood or urine, some of the sample handling or preservative methods are different for different types of chemicals or even among the metals. And that just increases the amount of sample you have to collect from people. I mean, that's one thing if people are in a hospital, but as you go to larger and larger populations and you don't want people refusing, I think we're going to have to try to be able to do it with a finger prick if we can, which probably will never happen. But I shouldn't say that. Anyway, I think also I'd like to caution that when you have a lot of things you're looking at, I guess it's always more important to collect more information. Just be careful about doing a lot of multiple comparisons, finding a statistically significant result, and then saying, aha, that's it, and then working backwards and then trying to a posteriori explain it. Versus, I think that's good for hypothesis testing, but then I think you need to design the experiments, repeat it, get multiple other experiments to validate that. With that being said, I think it's really important when you're collecting biomarker samples to have things like serum creatinine, so you can calculate what the glomerular filtration rate is in a person. When you have some chemicals that are not excreted very rapidly and you're measuring cholesterol, the two could be correlated just because of kidney function. Same way with urine, it's important to have urine flow rate if you have a biomarker in urine. So these are all things that I think people are going to have to really start looking at. What are the core things we also need to collect to look at different chemicals, different processes. And then as a toxicologist, I always like to plug, we need to know the mode of action to make any sense out of it. Thanks.
Okay, thank you. I think we need to talk about this for a little bit, about this, the whole issue of sample bias and so forth, because this is a major issue that we're going to have to address as we move forward. So comments on this?
Yeah, maybe I'll start towards the end of that conversation about, measurement of glomerular filtration rates, et cetera. So in the studies that I participate in, we just don't get samples and run them. We have lots of metadata associated with the samples, and I probably should have used the term nested when I was talking. I have a tendency to say case control studies when they're nested case control studies. I'm not an epidemiologist, I'm a physical chemist, so apologize if that came from me. So, so most of our studies, or at least studies in my core, are nested case control, and we have well-documented metadata. Particularly if it's any clinical study, it's like you want to know what is the gold standard that's being used now as the biomarker in the disease. And then we want to see if we use metabolomics, can we detect something earlier? Can we detect something? Can we detect something in baseline samples that are predictive of a health outcome later, et cetera? But all of these kind of measurements that are the gold standards are always provided with that metadata. And then we use those as the phenotypic anchors in our analysis, or we can use them to combine with the metabolomics data to say, can we get a better biomarker or a better predictor through making those combinations? But there was a first part of those comments also about the sample handling, if somebody, I mean, yeah.
I just wanted to touch on, you mentioned sample volume, analytical requirements, and I think this is where targeted, leveraging technologies and using targeted procedures can be very, very helpful. And soon you will see published where we did targeted GC tandem quad using 200 microliters of plasma, and we're able to see, TCDD, TCDF at 5 picogram per mil levels. So that's something we can do in children, and so there's room for innovation in terms of sample prep. That is a very rigorous sample prep. To do that is very, and I had to actually find a collaborator that could do that for me. And there's some of them. There's some in Berkeley. There's some in Pennsylvania that can actually do that. So there's room for innovation. And I think that we need to find ways to use smaller sample volumes to reach these very low levels. And I think when we use possibly this semi-targeted or blended approaches, if we can, where we're getting the bulk of the work done, via LC, electrospray, Hillick, reverse phase type technologies, that we also incorporate, we split off some of that sample to a very specific targeted. That particular assay looked at 6 persistent organic chemical classes at very low levels and covered a pretty broad range in terms of concentrations. And I think we can do that. So I just wanted to touch on, we don't need 80 mils to do that. We can do these things with today's technology with much less sample volume.
Okay, thank you.
Rishabh, I want to make sure that when we answer these questions that we don't go into the chemical-only sphere, because it's my thesis, having worked with chemical mixtures for 3-4 decades now, that if we knew the cause were chemicals or mixtures thereof, the solution is tractable. It may be very messy and expensive, but there are ways to get answers. To me, the integrative power of the metabolomics is the real power, because some of us have also been interested in things like the interaction both in timing and in susceptibility to things like exposure to viruses and exposure to chemicals. And you may have a wonderful dose-response curve in plaque-forming units, and another one that's wonderful in milligrams per kilogram per day. But it's almost impossible to decide what sort of metric or what sort of model to describe those two, much less if you put on a socioeconomic stressor, which has yet a different scale. And so the question I posed, and the reason I posed it, was because we have the possibility of looking at a variety of stressors at the point we're interested in, which is the body. But then the question of how you decide what your control is and how you identify the people who have the exposures, but it is not only the exposure, there's some other aspect, whether it's the genomics of the microbes in our gut or your original genetics, how that teases out, that would be very, I think, useful. But I don't think, I know, at least I don't know how to go about getting that from these data, because I think if we could get that information from these data, it would be very powerful.
Yeah, I think that's a very good point. I mean, we've already heard that diurnal changes, time of year changes. You put on top of that changes in the microbiome and the viral infections and so forth. It really is complex, and that's what we're going to. This gets back to that conceptual grid. How do we organize our thinking to be able to interpret this data? So, are there comments on that panel would like to make, or we can?
I'm not sure if I understood your question quite right, but here's something that, from experience, one of the best studies that I've done has been through patient pairs. And what it was a liver transplant study, and some of the patients after the liver transplant got fibrosis again, and so needed another liver transplant, so were basically dead. And then other patients didn't get fibrosis. So they matched the people, whether it was like a 20-year-old person and it was a female, they matched one that didn't get the fibrosis with one that did get the fibrosis, but they went through the whole thing together. Then they could pull things out and try to understand why the one was getting the fibrosis and why the one wasn't. And we got great correlation out of that. That was one of the best studies I've ever done. But it was having that perfect pair where everything was the same, the BMI was the same, the age was the same, the time they got the liver was the same, but it was just the outcome was different. That was one of the best studies I've done.
Okay. Yes, so I am Ana Navas from Johns Hopkins University, and I am also a committee member. So this morning it was quite interesting to see how the different speakers provided a different estimation of how much of the disease is attributable to the environment, from like around 50% to 90%. So that's a big difference. And when we think about how do we estimate population attributable fraction, we need to have good exposure assessment. We need to have high quality epidemiological research to be able to know how those exposures are related to disease. And so probably that's the reason why we have that variability and so much uncertainty of how much of the disease is related to environmental factors. And what I wanted to ask to the committee is, do you think, like a lot has been said about how much maybe the exposome, the metabolomics can help us to identify new causal factors that are important for chronic diseases. Do you think it can also help us to improve our estimation of the burden of disease that is related to already maybe known risk factors, so that pie that Steve Rappaport showed that had like, I don't know if it was a third or a quarter of already known causes for chronic diseases, and there was a lot of unknown. Could that part of the pie that is unknown be related to known causes, but that we have not estimated appropriately? And could the metabolomics also help us? So is it still useful to go for those known factors, or should we only focus on the unknown part of the metabolome? So that was kind of one of my questions. And then the other one is regarding we need cohort studies, that's obvious. And the question is, how do we do them? In the 20th century, in the last decades of the 20th century, we did amazing jobs of doing these population-based cohort studies with repeated samples. One of the best examples for environmental health I can think of is the normative aging study with lots of repeated measures over time. And the question is, those cohorts from the end of the 20th century, do we continue them? Do we build new ones? What are we going to do? This is a critical time right now in epidemiology in general, in clinical medicine, in health in general. How are we going to do the research of the 21st century? What type of cohort studies? For the US, it's critical. We have the UK example, other countries. With the medical records, there is not one single national health system in the US, so there are more challenges. So I think those are the things that we need to think about to connect with the metabolome. And then I have like one small comment about the personalized medicine versus population health, in the sense that we need to distinguish always between association and prediction. And when we look for associations and factors that could be causally related to disease, maybe they are not very good predictors. But we know that when we remove them from the environment, we intervene, we can actually contribute to prevent disease, maybe in some people, depending on how much of the attributable fraction is. But for prediction, sometimes in terms of personalized medicine, we don't care if that's causally related or not. It can help you to say you are likely to have a risk factor or not. We might not need to remove that factor. So I think those are the two things that I say.
Okay. So comments from the panel?
Well, I think to your first question, which is, that unknown part of that pie, I think that's where we should focus. I think that's what the exposome is about. We know a lot about that, smoking and those other known factors. So I think that our focus should absolutely be on those unknown things, and hopefully the exposome metabolomics workflows can help get us there.
Yeah, I agree with Anthony. I think that's the real power of the exposome concept is to do discovery. If we're interested in particular chemicals that are present in the environment, we can do targeted analyses now, the same way we've been doing them for years. If we want to use biospecimens rather than air measurements or water measurements, we can do that. The problem is we don't know, we're not good enough at guessing as to what of the 80,000 man-made chemicals that are out there are the ones that are likely to be most important. The geneticists weren't very good at guessing which genes were going to be important. It was only when we got GWAS and whole genome sequencing that we actually found it out. So, let's give ourselves a break here. And the exposome is really for discovery. And it gives us, instead of questionnaire data, it gives us quantitative, specific, precise information about specific exposures. And those exposures come from all walks of life. Everything that goes on in a living system is represented by those small molecules that are circulating in the body. So it's the beauty of it. It's a simplicity, but it's really for discovery. It's not for hypothesis testing. It's going to give us great ideas to follow up with hypotheses in the future. And we have to quit mixing and matching. We can't take the exposome and make it do both at the same time, because it's not what it's good for.
Yeah, just to add, I like how you said that. And it's interesting how plastics that were bad, or that they thought were good 5 years ago, then they find out are bad. So it's interesting what tomorrow is going to be bad for you that's maybe in your cell phone or something, and they're going to have to outlaw. So it'll be interesting to understand how the exposure changes throughout the years just based on the synthesis that they're doing to make the technology we use.
Okay, to stay on time, we're just going to be able to take one more question or comment. And
So I'm Christy Pullen from the Natural Resources Defense Council, and my question actually kind of builds on the comments that you guys just made. So one is about trends over time. So when you have that 1,000-fold difference between your xenobiotics and your endogenous chemicals, do we imagine that say, endogenous chemical line has shifted over time? So if you looked at samples in 1950 when we had less chemicals in the population, or 1900, or if we could go back to 1750, what would that endogenous chemical line shift? But then also, how do we think about the changing landscape? So things like nanoparticles, for example. So you might have, nanocarbon that's carbon. So how do you account for that in doing your metabolomics measurements? Maybe that's something that goes into the realm of changes that happen that you can't measure, the changes that are occurring in the blood itself, but how do we account for those shifts in what the chemicals are, so the new chemicals that are coming into play, as well as the chemicals that already exist in commerce?
I'll take the new chemical entity part. I'm very interested in that. And nanoparticles is one of the things that we work on in our group. But I think things like that are going to get detected through sensors that are for detecting the nanoparticles. But we've also been exposing animals to nanoparticles and looking at perturbations in the metabolome that are caused by nanoparticles. So we've done this with all carbon nanoparticles and also with nanosilver. And so it is interesting that, and actually some of these studies have been in pregnant animals and monitoring, that how that affects the pregnant animal. So we think about that as being how's that going to be transferred to the offspring? So there is research in this area. We certainly don't have all the answers, but I think the environmental detection and even perhaps the detection in tissues will probably be with the nanosensors, and then we can look at the perturbations and the pathways using metabolomics. And then we've actually made a lot of progress in using dark field imaging to look at the cellular localization of nanoparticles. So, it gets out of the metabolomics box, but metabolomics groups are probably sort of encompassing and looking at those things with the biomedical engineers. Yeah.
Okay. Well, thank you. So let's thank the panel for their comments and discussion.